Okay, let's get started. Quiz Monday, 7-1 through 7-3. That will take us up through citric acid cycle, which we are covering and will cover by Friday. So pages 135 through 142. I haven't given back the quiz, the last quiz, because a couple people still need to take it. But the quiz has been really good. I think the average is like 9 or something like that. So every quiz, the performance has picked up a little bit, which is good. We do have an exam coming up a week from Friday. That's going to be over cell respiration, and then the chapter preceding it, the energetics, thermodynamics, enzymes, etc. So 2 chapters, and so as I said last time, you really should be working on the first study guide for chapter 6, and then you should already be working on chapter 7 study guide as well. Questions? I know you have questions. You gotta have questions. I know Tuesday was probably a little bit overwhelming in some respects, because what I tried to do was kind of give you the overview and the big picture, and I really didn't expect you to understand how it all fit together the first time. What we're going to try to do is give you the big picture and then go back and look at some of the things in more detail, come back to the big picture again, and so we do this kind of alternating back and forth between looking at the forest and then looking at some of the trees within that forest. So, as a little bit of setup for what we're going to talk about here. What's the purpose of cellular respiration? To produce ATP, okay, and how do they do that? They do that by oxidizing glucose, and oxidation involves what? What do you do when you oxidize glucose? You remove hydrogens, okay, and as you remove hydrogens, you remove electrons. Where do those electrons go initially? Important point. What are the names of the enzymes that pull the hydrogens off of molecules like glucose? Dehydrogenases. And dehydrogenases need something to work with them in order to function. What's that something? Coenzymes, and very specifically, which form of the coenzyme? Yeah, NAD, the oxidized form. Okay, that's the form that picks up those electrons. Okay? Well, is there a lot of oxidized NAD in the cell?
No.
Now remember, coenzymes are made out of vitamins, like vitamin D is the one that's used to make NAD. And you don't need much vitamins in your diet because you don't have many coenzymes. So what's going to happen to your oxidized NAD during cellular respiration? It's going to become reduced. You're going to run out of it, right? And if you run out of your oxidized NAD, what happens to cellular respiration? Stops. And then we can't make ATP from glucose, so that's a problem. So a part of cellular respiration has to involve reoxidizing that reduced NAD back to the oxidized form again, or if you like, recycling it. Okay? What's the recycling mechanism in cellular respiration? Which part of it? Electron transport. And during electron transport, that NAD gets recycled. And during the process of electron transport, what happens across the inner membrane of the mitochondria? What builds up in this inner space out here? Hydrogen ions, right? And where are they low? In here in the matrix. And what do we use that hydrogen ion gradient energy for? Yeah, to phosphorylate, to make ATP out of ADP and phosphate. That particular process is called oxidative phosphorylation. called oxidative phosphorylation because the electrons, or this electron transport system, requires oxygen in order to operate. Now we're going to talk about the electron transport in more detail in a little bit, probably on Friday. But as those electrons are removed from reduced NAD, where do they go? Well, they go to this electron transport system. But then where do they go? They're transferred to oxygen. So oxygen is kind of the final resting place for those electrons. And as long as you keep oxygen fed to the mitochondria, this electron transport system keeps running. Reduced NADs can keep donating their electrons to it, and in the process become recycled. And the great thing is that during this recycling system, we set up an energy battery, if you like, that allows the cell to make ATP. Okay. Is oxidative phosphorylation the only way to make ATP? No, remember there's this other substrate level phosphorylation that doesn't involve all these membranes and electron transport. It just involves an organic molecule donating the phosphate to ADP. And that requires an enzyme. Okay? So those are some of the kind of highlights that you should have taken away from class on Tuesday. And if it didn't make sense on Tuesday, I'm hoping it makes a little bit more sense now. But if it's still not making sense to you, especially after we talk about some of the things today, you really need to come in and talk to me, talk to Austin, but you need to kind of get this straight before the exam. Okay? All right. So, this shows you kind of schematically the three phases of cellular respiration: glycolysis, citric acid cycle, and then electron transport. Okay? This particular slide illustrates a few things very nicely. Number one, it shows you the location of glycolysis occurring out here in the cytoplasm. And then it also shows you that the citric acid cycle, or Krebs cycle if you like, occurs in the mitochondria, specifically in the matrix. And then this electron transport and chemiosmosis is associated with this inner membrane of the mitochondria. So what we're going to do is we're going to go through each of these particular sequences with the mind of being able to answer these particular questions. Okay, so that's kind of our goal, is for each one of those particular sequences to be able to answer those particular questions. And those are the kind of things you will have to be able to answer for the exam. It's really key when we talk about these particular reactions right here to understand the organization of the mitochondria. Wonderful example of how the structure of an organelle determines its function. So this idea of this mitochondrial room, I'm going to keep coming back to over and over again, and you really do have to appreciate the fact that the mitochondria has an outer membrane, cytoplasm out there in the hall. It has an inner membrane, these outermost row of chairs all the way around the perimeter of the room. That's where the electron transport chain is going to be. It's got a matrix, kind of a cytoplasm-like fluid inside the mitochondria. And then it's got this space between the inner membrane and the outer membrane. So a space, a membrane, a space. That allows us to make that hydrogen ion gradient to kind of charge that battery that's used to make ATP by chemiosmosis. Okay? So these two particular slides right here, which are pretty small, you can see them if you print out slides, shows an electron micrograph of a mitochondria, and then down here a conception of it that shows the outer membrane, this little space in between the inner membrane space, the folded inner membrane, and then the matrix inside here. And remember that because the electron transport chain is in that inner membrane, and also those channel proteins that allow the protons to flow through, which is an ATPase enzyme, the more folded this membrane is, the more surface area there is, And then the more complexes there are, electron transport, ATPases, to make ATP. So greater surface area, you can make more ATP. And this particular illustration right here just shows the same thing. It's a little bit different slice of the mitochondria, showing the folding of that membrane. And then down here, this is a micrograph of the inner membrane. And you can see these little kind of bead-like things stuck on the membrane, higher power here. Those are actually those ATPase channel proteins that are embedded in this mitochondrial inner membrane. So structure determining function is just really well illustrated with the mitochondria and cellular respiration. Okay, so we're going to start with glycolysis. Glycolysis begins with a glucose molecule. What's the molecular formula of glucose? C6H12O6. Okay, so it's got a lot of hydrogens. Hydrogens equal potential energy. And during the process of glycolysis, we are going to take one glucose, which has 6 carbons, okay? And we're going to convert it into 2 pyruvates, each of which contain 3 carbons. So we're basically going to split the molecule in half. During this particular process, there is going to be an input of ATP required. Remember I talked about that a little bit last time, priming the pump and sticking some energy in so you can get more out. But when the process is over, we're going to have produced 4 ATP. So what's our net per glucose by glycolysis? 2. Okay, 4 minus 2. And how are these produced? By what process? Oxidative phosphorylation, or is it substrate level? It's substrate level. And how do you know that? Because it's happening in the cytoplasm. You do oxidative, you've got to have a membrane in the mitochondria. Okay? So we make 4 ATP by substrate level. During this particular set of reactions, 2 oxidized NAD are going to become reduced. Okay? So someplace along the way, we're going to pull some hydrogens off of glucose and transfer them to NAD. Pyruvate is going to have less hydrogens on it when we're done. One of the questions that you're asked about each of the phases is carbon dioxide produced? Is carbon dioxide produced during glycolysis? Can't be, can it? We haven't lost any carbon. We've just basically taken a 6-carbon molecule and chopped it into 2 3-carbon molecules. Can the cell continue to just do this indefinitely? Can it just do glycolysis?
Some cells do that, don't they? They only do that.
There is no cell that can do just glycolysis. Why not? Okay, it takes 2 ATP, you're right, but you do produce 4, so you would get a net of 2. Why can't you do just glycolysis?
Wouldn't all the energy just go into glycolysis and not other cell functions? There wouldn't be enough ATP to do anything else.
Okay, well that is a pretty paltry amount of ATP. But as it turns out, there are cells that can survive quite well and flourish, only producing a net of 2 ATP per glucose. including E cells. What's the problem?
Pyramid,
Pardon me?
the pyramid.
Is there something about pyruvate building up? That's a possibility.
They convert it to ethanol.
Okay, we're getting there. We're getting there. But what is the limitation that makes this impossible to happen indefinitely?
There won't be enough NAD.
You run out of this, right? You take all of your oxidized NAD and reduce it. So we're going to see what the strategy is in just a second. Okay, so this is going to break it down, glycolysis, into a little bit more detail. And it's going to show you some of the kind of intermediate steps. I'm showing this to you primarily for illustrative purposes so you can see the logic of what's happening. But we have addressed most of the things, if not all, that were on that master list that you need to know about glycolysis already. Okay? Is ATP used? Yes. Is it produced? Yes. How much? Is NAD reduced? Yes. Is it oxidized? No. Where does it happen? Cytoplasm. Is CO2 produced? No. Okay? So some of those basic questions. Okay, so here is glycolysis from an energetic standpoint, kind of an energy ledger. And remember, one of the things that I'm going to try to do in this particular section of cellular respiration is have you come to an appreciation for kind of an energy budget for the cell. We know that under ideal conditions, aerobic cellular respiration can produce how many ATP per glucose? Remember that number? 38, okay? So we're going to want to kind of see where they come from.
I have a question about the oxidation of NAD.
Okay?
Wouldn't the first part of it be oxidized and the second part be reduced? Isn't that what you said earlier?
Okay, here, if we add electrons to the NAD, then the NAD does become reduced. At some point in time, we've got to pull those electrons back off again, so this will become reoxidized. But that doesn't happen in glycolysis. Okay? So it's going to have to happen someplace, you're right. But it doesn't happen during glycolysis. Okay? Okay, so here's our glucose. And very early on, there is this energy investment phase of glycolysis, where we've got to stick a couple of ATP in the process to get it going. So energetically, okay, free energy of glucose is up here, free energy of pyruvate is down here. So what kind of a reaction is this? Hexergonic or endergonic? Glucose, pyruvate. It's hexergonic, right? But in reality, one of the first steps actually is up here. It's higher than glucose. That's endergonic, right? We've got to stick a little bit of energy in to begin with, and then we get a payoff later on. So we use up 2 ATP. We form 2 ADP and phosphate. Now, in some of the later reactions, we get our energy back. We take 4 ADP and we phosphorylate them by substrate level phosphorylation. So we get 4 back. Okay? So a net of 2, positive 2. And then in some of the reactions, which are oxidations, we strip hydrogens off of molecules and we donate it to NAD. And in the process, NAD becomes reduced. Okay? So here is kind of our net for glucose, for glycolysis. We use the glucose, we convert it into 2 pyruvates and some water. We make 4 ATP, but we used up 2 for a net of 2, and we reduce to NADH. Now this is a problem. Okay? This happening just by itself is a problem. We're going to run out of this. But this represents an opportunity, because later on, if we can recycle this, especially using this electron transport chain, then we can make ATP. For every reduced NAD that is oxidized by the electron transport system, we can potentially get 3 ATP worth of energy out of that. So this theoretically, if we have some other processes occurring, represents 6 potential ATP. We're not there yet. We haven't gotten yet. But this has the potential energy to make up to 6 ATP. Okay? Okay. Now we get into the real treats. And again, this is stuff that I don't want you to obsess over or to worry about having to memorize. I just want to show you what's happening so you have some kind of an appreciation for what's going on within that black box, if you like, of glycolysis. Okay? So, here's glucose, C6H12O6. And the first reaction of glycolysis involves the conversion of glucose into glucose 6-phosphate. So we phosphorylate glucose. See that phosphate group right up here? Where does the phosphate come from? ATP hydrolysis. This particular enzyme that catalyzes this reaction is called hexokinase. Don't worry about it. Don't worry about the name. Okay? The next step involves an isomerization of glucose 6-phosphate into kind of a cousin of glucose, fructose. So basically this enzyme, phosphoglucoisomerase, converts this molecule into this. Okay? What's the next step? Another phosphorylation. So we spend another ATP. And what do? We stick a second phosphate group onto this, forming fructose 1,6-bisphosphate. Okay? So we still have a 6-carbon sugar, but now after these steps, it's got a phosphate group on this end and a phosphate group on this end. Haven't gotten any energy out yet. But we've expended 2 ATP to get to this point. There is now an enzyme called aldolase that can convert this molecule into either this or this. These are 3 carbons. This is 6 carbons. So basically, we split this molecule apart. And sometimes we form this, and sometimes we form this. These 2 molecules can convert back and forth to one another. There's an enzyme that catalyzes this called isomerase. This one, this glyceraldehyde 3-phosphate, is the one that's going to eventually be converted into other things. And as we use this up, this will be converted into this. If, again, that detail is a little bit too esoteric for you, don't get hung up on that particular detail. So far, what have we done? We've taken a glucose at the expense of 2 ATP, involving several sequential step-by-step reactions, and we basically converted glucose into 2 molecules of this glyceraldehyde 3-phosphate. Okay? So where do we go next? The glyceraldehyde 3-phosphate is now oxidized into this compound. This particular compound, oh, I'm sorry, we missed a step. Okay. This compound now is oxidized, and in the process, hydrogens are removed from that compound, forming this compound. And in the process, we make reduced NAD. So we get our first payoff now of reduced NAD. 2 molecules of reduced NAD are produced. This molecule now is dephosphorylated. We remove one of the phosphate groups, and we use that to make ATP. That produces a molecule now with 1 phosphate group. We slightly rearrange this molecule a little bit. We remove a little bit of water from it, and then we remove that additional last phosphate group finally forming 2 additional ATP, and now we're left with pyruvate. Rachel?
Is this happening with the 2 compounds from the last time at the same time?
Yeah, well, not really. Not really. What really happens is it's really happening to this one, and then this one is being converted into this one. So we're really following what's happening to this. What's a little bit confusing and makes this a little bit confusing is you've got to remember that 1 molecule of glucose actually produces 2 molecules of pyruvate. And so when we do our accounting and you give these numbers, we're really accounting on the basis of what energy do you get from a glucose molecule. Now again, I almost in some respects hesitate to show you this because you become all kind of confused and consumed by all these details. But I think it is important to show you the step-by-step reorganization that takes place, because this is relevant when we talked about metabolic pathways. Remember you said in metabolism, something is converted into something else, converted into something else, something else, something else. So the process of glycolysis is not just bang, glucose to pyruvate. It's a step-by-step arrangement involving little minor adjustments to the molecule, little tweaks, adding a phosphate here, taking it away, removing some electrons, donating to reduced NAD, and ultimately we get down to this molecule. Is pyruvate more oxidized or more reduced than glucose? It is more oxidized. How do we know that? Very top here. We took hydrogens off of it to make the next step in the molecule. Where do those hydrogens go? To reduce NAD. So glucose had more hydrogens than pyruvate had. Some of those hydrogens end up as part of the reduced NAD. Is there still energy in this molecule? Yeah, there is. They're not showing you all the little hydrogens that are still attached to these carbons. So there's still hydrogen on this. There's still potential energy in this. There's still calories, if you like, within this molecule. Now, if we want to further process the pyruvate in the mitochondria, oxygen's got to be present. Okay? Remember that glycolysis occurs in the cytoplasm. So in order for this pyruvate to now move into the mitochondria and participate in the citric acid cycle and the electron transport chain, that's not going to happen if oxygen's present. If oxygen is present, then we're doing aerobic cellular respiration, and then we start getting more energy payoff as we continue to break down that pyruvate molecule. But also remember that we can't stop here. Okay? Why? Because we would run out of this. This enzyme wouldn't work. And therefore we would miss these energy payoff steps. Unfortunately, these energy requiring steps would still occur. So we would chew up energy, but then if the reaction stopped here due to the lack of oxidized NAD, we wouldn't get the energy payoff. Okay? So, in lab, you worked with yeast. And we were doing an experiment to show the rate of carbon dioxide production during this process that we called fermentation. Does fermentation utilize oxygen? Is it the same as aerobic cellular respiration? No. What is the payoff of fermentation? It still uses 2 ATP. It still produces 4. So the payoff from fermentation is going to be plus 2 ATP, just like glycolysis. So it doesn't get us any more ATP. Fermentation consists of glycolysis plus some extra steps. What's the purpose of those extra steps in fermentation? Well, remember what glycolysis produces. It produces 2 reduced NADs, right? See them up there? What's the purpose of these extra steps? To convert the reduced NAD back into the oxidized form. The purpose of these extra steps is to recycle this coenzyme. Now, depending upon the type of cell and the type of organism, these additional steps that are tacked onto glycolysis can vary from cell type to cell type. Yeast, for example, carry out a type of fermentation called alcoholic fermentation. And what is the end product of the fermentation process? Obviously alcohol, right? So in yeast, fermentation consists of glycolysis, and then the pyruvate is ultimately converted into alcohol, ethanol. Why? So this can happen. A lot of bacteria and your muscle cells do a different type of fermentation. If you go out and run a lot and put yourself into oxygen debt, so your muscles can no longer be sustained by oxygen, then the aerobic cellular respiration stops. Can they still produce energy? Yeah. They can ferment. Do your muscle cells do alcoholic fermentation? Kind of interesting, wouldn't it? No, they do a type of fermentation that some bacteria can do as well, called lactic acid fermentation. So they have a different set of reactions tacked onto glycolysis. And those produce lactic acid. What's the consequence of lactic acid buildup in your muscles? Soreness, okay. Why are these extra steps occurring in lactic acid fermentation? To do this. It's all about doing this. Do you get any more ATP out of this process? No. But what does this allow you to do? To keep glycolysis going. If we didn't have these extra steps to do this, we would run out of the cell's oxidized NAD. Does that make sense? That's a really important part of understanding cellular respiration and fermentation. Okay, so, whoops. So here's our glucose out in the cytoplasm. Sorry, cytosol. This was out external to the cell, so the cell's going to transport some glucose into the cell by a facilitated diffusion pump. And in the cytosol, we now convert glucose into pyruvate. If oxygen's present, the pyruvate can continue to move into the mitochondria and participate in aerobic cellular respiration, citric acid cycle, electron transport, and get all those 38 ATPs out. But if no oxygen's present, we can keep glycolysis running if we tack on these couple of extra reactions to it. those recycling reactions. It doesn't get us any more ATP, but we can still get that 2 net ATP per glucose that we got out of glycolysis. And for a simple little yeast cell, that can be enough energy to keep it live and going. How about for a brain cell? Not enough ATP. Okay. Brain cells, nothing. As a product of fermentation, we can produce ethanol, we can produce lactic acid. There's all kinds of other types of fermentative pathways, particularly in bacteria. We can produce butyric acid. Butyric acid is responsible for the smell of rancid butter that's spoiled. Bacteria are fermenting the sugars in the butter and producing this butyric acid by fermentation. Smells like really nasty. We can produce acetic acid. All kinds of different things can be produced by fermentation. So, here is alcoholic fermentation. Here's our glucose being converted by glycolysis into 2-pyruvate. During the process, we make this net of 2 ATP, and we are reducing NAD during glycolysis. Pyruvate being converted into ethanol is the recycling process. During this process, we're actually going to be converting the reduced NAD back into the oxidized state again. This is going to have more hydrogens than this does. Where do they come from? They come from here. The first step of alcoholic fermentation involves converting pyruvate into a compound called acetaldehyde. I don't care if you remember this name. Okay. What I do want you to see, however, is that pyruvate has 3 carbons. Acetaldehyde has 2. So for every 2 pyruvate that are converted into acetaldehyde, we form 2 molecules of carbon dioxide. That's what you were measuring in those tubes, or will today.
Where does the hydrogen come from?
Which hydrogen?
The one connected to the carbon pseudoaldehyde.
Oh, they're not showing, it's unfortunate, but they're not showing all of the hydrogens on here. They're kind of abbreviating these molecules. Okay. They're showing this one for some reason because it's on the end of the carbon. So, this process does evolve CO2. Acetaldehyde then has hydrogens added to it from here. Well, that's the source of one of them right here, from NADH. Okay. Has hydrogens added onto it, and then is converted into ethanol. So ethanol is more reduced than acetaldehyde. The source of those hydrogens comes from the reduced NAD produced during glycolysis. So basically, this hydrogen, Jesse, comes from here. Okay? 2 of these, 2 molecules of this, 2 molecules of this. Now, it shows you in some respects how desperate yeast cells are to produce a little bit of ATP by fermentation. Because what is this to the cell? What is this final product of fermentation that actually accumulates in the cell? It's a toxin. Okay? And if cells build up too high of an ethanol concentration, it'll kill them. That's why you can't naturally ferment wine over an alcohol content of about 13%. That's the maximum you can get from wine. If you ferment mash and things like that in making higher proof liquor, the first step is a fermentation, but again, you reach your alcohol limit, and then you have to distill that to drive off some of the water to make a higher alcohol content. So these cells are basically potentially poisoning themselves to make that little bit of energy. Okay, here's lactic acid fermentation. So again, here's our glycolysis, our 2 ATP that's made, the 2 NADs that are reduced here. Here, pyruvate is converted into lactic acid or lactate. This has got 3 carbons. Pyruvate's got 3 carbons. Okay? But again, this has to accept hydrogens to be converted into this. That is provided by, those hydrogens are provided by the reduced NAD. So what determines whether you can do this or this? It's all about the enzymes you have. Okay? Our muscle cells do not have the enzymes to convert pyruvate into acetaldehyde into ethanol. Why? Because we don't have those genes. Muscle cells and bacterial cells, at least some of them, have the enzyme that can do this. Why? Because they've got the genes. So the end products of fermentation are determined by what genes as a cell you possess, because that determines what enzymes you can produce. This one's kind of hard to read, but this kind of shows you glycolysis and fermentation with names of compounds stuck in there. Again, I'm not interested in you memorizing all of these names right here. For glycolysis, you need to know the starting product, and you need to know the end product. But this shows you kind of step by step what's happening in each of those conversions. And then once you've got pyruvate, depending upon whether, I think this is showing ethanolic fermentation, it's showing the final steps of fermentation and what your energy payout is at various places. So this is just a summary of that. Well, again, 2 ATP from fermentation is not much. Certainly isn't 38. Ideally, oxygen will be present, and that will allow the pyruvate to enter the mitochondria and participate in the citric acid cycle, and then also for this electron transport system to function. Because right now, we only got 2 ATP produced. So let's take a look and see what happens at the citric acid cycle. This particular reaction right here is oftentimes referred to as the transition reaction. And basically what this is doing is this is preparing the pyruvate to enter this citric acid cycle. When I account for what's happening in glycolysis, citric acid cycle, and electron transport, I lump the transition reaction in with the citric acid cycle. So I consider this all to be part of the citric acid cycle. So let's just take a look at the transition reaction, and then that's a good place to stop. And tomorrow we'll look at the citric acid cycle. So what's happening in the transition reaction? Okay, the pyruvate is moved into the mitochondria. And particularly, it has moved into that matrix of the mitochondria. So that pyruvate that was produced in the cytoplasm has moved into the mitochondria and is now sitting here. And this is where all of the enzymes of the citric acid cycle are located. Well, there's a very, very complicated enzyme complex that works in this transition reaction called pyruvate dehydrogenase. Again, don't have to memorize that name. This particular enzyme complex does a lot of things to pyruvate to get it to the point where we're now ready to enter the citric acid cycle. So in the transition reaction, pyruvate is converted into this molecule, acetyl-CoA. Well, look what's involved in that happening. Pyruvate has 3 carbons. Acetyl-CoA has 2. So we're going to lose a carbon in the form of carbon dioxide in this reaction. This has more hydrogens than this does. So we're going to have some hydrogens removed. Where do they go? They go to NAD. So we reduce NAD in this step. And then temporarily, a coenzyme. Remember, coenzymes usually partner with enzymes. But here, we temporarily stick a coenzyme onto this molecule. And one big enzyme complex does all of these kind of simultaneously. Remove hydrogen, remove carbon, stick on coenzyme A. And in the process, we form this acetyl-CoA. Now, from an accounting standpoint, remember, a glucose makes 2 pyruvates. So for every glucose, we make 2 of these. We produce 2 carbon dioxides. We use up 2 of these acetyl-CoA molecules. Once we've got this, the remaining steps of the citric acid cycle are all about breaking this further down into more carbon dioxide and getting some of the chemical bond energy out of this in the form of ATP and some reduced NAD and another coenzyme, reduced NAD. So we'll dig into that a little bit more next time. So at the beginning of the period, I'll kind of step back again and kind of reevaluate and say, okay, big picture, where are we? What should you know? Where should you be comfortable at this point? You can do a little bit of review of glycolysis, fermentation, and then we'll add on the citric acid cycle. Okay?